describes your weekend? What is it? One word or one thing? Boring, chill, relaxing, none. Does that mean you had none weekend? Gifts. That sounds fun. Laying, traveling. Draven, where were you traveling? Fun. I like that, James. Cats. Oh, sick. That's not fun. How are you feeling today, Naya? Any better? Not that that's a fun way to spend your weekend. Traveling. Oh, that sounds fun, Amber. I'm trying to get back. Sorry, I don't want to the top of chat and working. Oh, spent the whole day having fun in Cleveland. That is fun. We had all kinds of weekends, sick weekends where we were working weekends where we were just being chill and lazy. Family. That sounds so fun. I always like being sick on weekends because I feel like then you have time to recover. You know what I mean? Like you have all these things you're expected to do, but then at the same time, that's like kind of like when you like to have like fun rest and do fun things that restore you. So it's like that catch-22. You know what I mean? You didn't have to try to like work. My daughter Mila, I'm pretty sure we have family that had actually and friends that had influenza A and influenza B. I'm pretty sure she had that Friday, and I just kept pushing through working with her on her school for OHVA, but she was just laying on the couch, and I was just going through her OLS with her in the afternoon. Yeah. So, snowboarding, that's fun. Fun way to embrace the cold weather. It's also nice when it's cold and icy out. We got snow Friday. So, it's nice when you can just stay in, right? Well, I'm thinking of all of you who are sick. I hate that for you. I'm sorry that some of you had busy weekends, but nonetheless, I'm glad that we're here together because this is one of my favorite things to do is just be with you all. So welcome back to biology. It's Monday. It's a fresh start. So I always like Mondays. I don't like to think of them as the start of something I don't like. I like to think of them as a fresh start. So whatever your week was like last week, know that this week doesn't have to be that way, right? We get a fresh start on Mondays. So let's start our fresh start out good. I'm starting mine out with fruit and coffee. And so that starts where we are today. I have several of you that are like, what is all these weird combinations going on the board? Let's start down here in this bottom corner with the banana. All right. Before we get into what we're doing today, I thought we need to get our brains going. All right. Because it is a different topic that we're going into today that kind of can feel like it's unrelatable. All right. So this banana. All right. What is keeping me from eating this banana right now? What's keeping me from eating it? I like the way you're thinking. The peel. The peel. So what do I have to do if I want to consume this banana right now? I got to peel it. Okay, I'm not going to peel it because I don't want to eat it during class. I won't be chomping on it during class. And if I open it, it'll become brown. But we know what it looks like if I would crack this open and peel the banana, right? Yes. So the only thing that's keeping me from this banana is to peel it open and then I can eat it. What about my TV? My TV is up there. Okay, my TV's up there. I have a TV and a remote. What is keeping my TV from turning on to the Today Show? Like, why what's keeping me from watching the television right now, Getting some good Jenna and Hoda in. All right, the powerhouse school. School, yes. But yeah, I could have this on if I would just push the power button, right? It's like I'm so close. Like my finger is on the remote. All I'd have to do is apply a little bit of pressure and that power would turn on to the TV. The news is on. Yes. I like to watch the news too, keep myself informed. All right, similar. All right, if and I don't have mine with me right here, have my headset and my computer, what is the thing that needs to happen to get that going in? Yes, Alexis, the plug, right? If I have my headsets on, but they're not plugged in, then I'm not going to hear anything. But if I plug it into the side of my computer, apply a little pressure and get that sucker in where it's supposed to go with metal on metal, then it's going to translate that connection or the Bluetooth. Good. Yes. What about I have a refrigerator that has a ice maker where you put your cup on there. I have a cup and I've got my refrigerator door that I can get ice out of it. I want ice. Right. I don't just put it underneath there. It's not censored with like a visual sensor. I have to push on that, right? You push it on that little cup holder piece and then the ice will fall out. We're good. All right, so this is kind of where we're going today in biology. So we talked last week about gene expression, right? We talked about our eyeball and some things that our eyeball cells do not need. What are some things that our eyeball cells have DNA in their eyeball cells for, and our eyeball cells for, but they don't do that or use those things? Good. Yes. So they have digestive information, such as like how to produce the digestive enzymes or proteins that our stomach cells and our intestine cells need to digest food. Our eyeball cells do not need that acid constantly being produced by their cells, right? I mean, that would be crazy if you could just lay a piece of turkey on your eyeball. Yeah, that would hurt. That would be bad. We don't need those kind of proteins being produced by the cells in our eyeball. Do our eyeball cells have a complete set of DNA that could tell our eyeballs to produce that? Do they have the information? Do they have the recipe in their eyeball cells? Yes, you're right. Yes, 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 yes. You guys are so smart. Yes. All right, they have. If I got an eyeball cell and pulled it out and unwound that DNA and looked at it, that it has a complete set of how to do all of those things. But why don't we produce everything that we have a code for? Because our cells differentiate, right? We talked about that kind of being like going to a trade school or a college. They get specialized as they get assigned a job. They're like, you're going to go be this kind of cell, and they go get specialized. And we talked about this wall behind me being full of light switches, right? And that some of that DNA, like this, how to produce stomach acid, is turned off because it's not information we need to know, right? Kind of like my algebra and trigonometry things that I've learned in math, like how to do complex equations or whatever else. I don't use those as often as I did when I was in high school and college. And so some of those things aren't super fresh on my mind, right? But what about the stuff that I learned in high school biology? Tell me what happened to them. They are relevant, right? Like, you need them, but like I need them even more because I went into science. So that stuff, not only is it basic, but I also took in high school, I went on and took like an AP, a college credit biology when I was done with biology. I loved it so much that I went on and took college credit classes in higher level sciences. And yes, I do still use them every day because my specialized area was science. And I especially use them because I teach you guys those things that I learned in high school biology today. I think science is fun too. So today we are going to learn a word that is totally weird. All right? Totally weird. That has to do with how our genes get specialized and how sometimes that DNA that's in there, those genes that are in there, the recipes for things that are in there get turned off, right? Just like that light switch. All right? How does that happen? And we are going to look at that today a little bit closer. And I'm going to tell you, I like to prepare your mind. This is not going to be impossible to understand. It's not. Because we did these things, right? We've got our brains prepped for what this is. And we're going to use a word called the lac operon today. Can you say lac operon? We are going to sound totally sciencey. Everybody say lac operon. Does anyone want to get on the microphone and say lac operon?
Lac operon?
I have no idea.
Yes. Thanks for jumping on there. Lac operon. Sounds French. Dude, you guys are going to sound so fancy today. If your parents are like, did you do your school? What did you do? Tell me something you did today. They'll be like, I can tell you about the lac operon. Or just name drop the lac operon and you're going to sound fancy. All right. So we're going to learn about the lac operon today. Before we read the goals, I want you to tell me about where those goals are. Where are those goals on our level of understanding of the triangle? Top or bottom? Look at the color. Remember, look at the color. Yes, they're at the bottom. So that tells you today this is fundamental learning. All right. When you leave today, you shouldn't be able to take this apart and re-put it together and explain to someone in a project how to do this. Today we're on that basic core level building, right? We're going to build the pieces of what this means today. So don't stress yourself out trying to feel like you're up here. We're down here today. We're going to get the basics. We're going to feel strong on the basic knowledge of our content today, and then we can build, okay? Who wants to read our goals? Luna, you want to read our goals? Anybody else want to read today before I start assigning? All right, so Luna, I'm going to have you read the first two, and Sadie, I'll have you read the second two goals for the day.
All right, do you want me to read the first two or the entire thing?
The first two would be perfect.
All right. What is lac operon? Where is the lac operon turned on?
Perfect. Thank you, Luna. All right, Sadie.
When is the lac operon turned off? What causes the changes from infant to adult? Thank you.
Thank you. All right, tell your classmates how awesome they are for getting on the mic, getting on the cams. Yes. Perfect. All right, if I had to ask you what you know about a lac operon right now, give me the emoji that would explain what you understand about a lac operon. Don't try to impress me. It's okay. You don't have to feel like you know what's going on. We've got more smiley faces. I was anticipating 0. I'm going to be honest with you. Not too many people could talk to me. I want to say a smiley face means you could tell me right now what the lac operon does or answer some of those questions. Maybe you know. I'm impressed. More smiley and mez than I anticipated. This is how we feel about it. All right. Awesome. So let's go first. I would like to take us to a little movie about gene regulation from the only person that I would have you watch a movie from. The only other teacher that's better than me. No, I'm just kidding. Mrs. Palmer. No, Mrs. Palmer is a better no. Mrs. Palmer is great, but what I was talking about is the Amoeba Sisters, right? We love the Amoeba Sisters. They're funny. They have great pictures. Oh, no teacher is better than me. Oh, that's sweet. But the Amoeba Sisters just have it. In fact, they are my role models. If I could be someone other than myself, I would be an Amoeba Sister. They have the best way of simplifying complex processes and pictures. Who is it? Here we go. This is where we're going to do. We're going to take a minute and watch this. We will not watch the whole thing, but I want to get just start here. Let's start here. All right, I'm going to play this. Let me see. Yes, I'll put the link in chat as well so you have that. All right, let's play this and then I want to make sure you can hear it. Time out. Are we made of prokaryotic cells or eukaryotic cells? What do you think? Do we remember? Dig back in. What does U mean? EU equals what? It rhymes. U equals, yes, Jenna, high five, true. U equals true. True what? Eukaryotes have a true. Yes, nucleus. They have a true nucleus, a membrane-bound nucleus. Remember the fireproof safe inside your cell that has all the DNA in it. Are we made of eukaryotic cells or prokaryotic cells? Yep, we are made out of eukaryotic cells. Good. Prokaryotic cells were like the first cells. They're the most simple, right? They still have DNA, but they don't have that membrane-bound nucleus, that fireproof box safe. The DNA is just throughout the cell. All right, let's continue. All right. Do you see what we're talking about with the block? What is the block in the banana example? The peel, right? The peel. The block in the remote is actually, you got to push it, right? This is where we were going. So let's keep listening in. Think about that promoter and that operator. Really, like, this is something you can be drawing pictures of as this goes on, or writing notes as it happens. Does that make a little bit of sense? Yes, Lizzie. Yes. So Lizzie is lactose intolerant. So we get to talk about something that is super relevant in your body. Your body blocks that mRNA from being produced. Yes. So you're a precise example of this exact process. All right. That there are those repressors in our body that keep things from being produced. Now that can be a bad thing or a different thing when you talk about like lactose intolerant, right? Because your body doesn't produce what you need to break down those things. So when you don't actually have anything to pull that repressor off, yes. So, or that it can also be a good thing. Like we started talking about, like with the hydrochloric acid in our stomach, sometimes we don't need those things to work. So them not being turned on is really okay. So the lac operon is not the only thing that has the repressor. All right. This is an example. The lac operon is one example of how this is used. All right. So what happens when someone is lactose intolerant, right? They don't create the enzyme lactase. So your bodies can't break down the lactose in dairy products. All right. When it was talking about bacteria, all right, E. coli is a bacteria that lives in our intestines, right? We often think of bacteria as bad guys, right? That our body needs to fight off. But we also have bacteria that are good guys, and they live in our intestines, and they're important because they help us break down food. All right. And that's the case in this situation. How do they know when to get working and creating lactose? Well, they know that because lactose is present, right? They can't start that process until lactose is present. That's how they know there is a need. So let's look at this from a specific standpoint. All right. This is a strand of DNA. We're looking at a piece of DNA here. All right. So here is the promoter. That's the guy that kind of flags down the site in the DNA where it's like, hey, the code starts here, right? That binding spot. So the promoter, they had like what did they show the promoter as, like a cheerleader? Like here's the beginning. All right. And then that operator right next to it is actually the go point. All right. It's actually the point that it sends it either moving on to produce that code or where it's blocked. And it says, you don't need to do this right now. This isn't relevant to your job, or this isn't relevant to this situation, right? So like in the lactose example, lactose isn't present. You don't need to create this. Does that make sense? To this point, does that make sense? You have the operator or the promoter that's like the flag, the starting point. Just like in a construction zone, you don't just drive into a construction zone, right? There are signs that say construction ahead. That would be like our promoter. Construction ahead. And then the next thing would be what? You'd actually start seeing those codes or a lane closure or whatever it is. Good. Most of us are there. If you are one of the few that said you're not quite there and getting this part of the process yet, private chat me. Where's the roadblock? What is it specifically that you're struggling with? We've got the DNA. The promoter is the flag that says start copying here. And then the next piece is the part that says you can begin, right? So if I get this is the banana. It's telling me like this peel. It's telling me, okay, this is where the banana begins, but I can't actually eat the banana, right? The operator is the peel. Because it's yellow and the peel's still on it, it's like the repressor. The peel is the repressor that goes on that operator, and it says, too bad, can't have this right now. But when I crack this banana open and peel it back and it's white, it is like the operator saying, come ahead. Have you ever been in a car wash that you drive through? Yeah. All right. So like you drive up to it and there's a sign, right? An entryway where you drive in. That entryway is like the promoter. This is where the car wash is. This is where it starts. It's a sign, right? But then you drive up to get in there and there is a sign that says stop. I mean, you don't drive in there if the sign says stop, right? Because if you drive onto that belt too soon, you're going to be hitting somebody or you're not going to be getting a full car wash, right? So once that stop sign says go, that's like the repressor is lifted. The operator is like the one that starts that red light, green light into the go process of the car wash. It's the same way in our DNA. The sign for the car wash and entrance is there. That's the promoter. And then you drive up to the car wash entrance, start tunnel, and if it says go, then we can produce whatever that DNA is coding for. If the repressor is on it, the stop sign's up and it's saying, you don't need this right now. All right, does that make better sense? Let me resend that out. Does that help? See where we are with that? Good. So it's our DNA. It's in every cell. And there are these little signs for all the different things that are going on in our DNA, all the different things we can need. There's signs, just like there's restaurant signs, there are gas station signs, there are car wash signs, there are shopping mall signs and hospital signs. Think of all the signs you see on the road to tell you where something is. That's what's going on in our DNA. The signs tell you where it is. And then on almost all of those places we mentioned, on the door, it tells us what? Whether it's open or closed. Yeah. Directions. Go this way. Come on in. I can't pull up to Subway at 2 a.m, right? If I walk up to the door at Subway at 2 a.m. and pull open the door, am I going to be able to get in? No. It's going to say closed. You don't need Subway right now. Go to bed, right? You're right. Now, Gracie, if I went to Taco Bell, they might be open, or Waffle House, right? But that closed or open sign is just like that operator. And I love that we use the word operator because the operator makes sense to me. It tells us whether it's open or closed, whether you need it or you don't right now for the type of cell you have. Now you want Subway? Sorry. Oh, all right. So this is another picture of it. All right. Again, this is just another way of showing you the same thing. DNA. This is the repressor. When the repressor is on, that RNA polymerase can't slide through and do it. I also like to think of it as like a bottle lid, right? I like to this spigot, right, is the promoter, is the sign. It says water is here, right? And that nozzle is the operator. They both have open nozzles. So it's like the water can freely flow and the protein can be expressed. But if the bottle lid is on, can water freely flow in? It's a block, right? Right. It can't do that. Another example, I stole this from Ren today. No. Hold on one second. Let me grab it. Tell me if you ever had a marble run when you were younger. Did you ever have a marble run? Okay. I'm going to do my best to set this up for you. Does everybody see the marble run? Yes. All right. So up here at the top is the little bead. This bead represents, like this bead, it represents the RNA polymerase. It's the enzyme that's going to make your mRNA or your copy of your DNA that goes to the ribosome and makes your proteins, right? So it's there. It's found the DNA that it wants. It wants to copy this strand. All right. The strand is represented by the marble run track. But what represents the repressor? It's saying, no, you don't need this right now. Don't break again. Yes, this right here is the repressor. It's keeping my bead from marble from traveling down. If you look, there's a little pocket in the track, right? That marble sits in. I like to think that of like the promoter. It's like, sit right here. This is where it begins. If you even look on the track, I'm going to pull it apart for a minute. There's an arrow right here. Do you see that? There's an arrow to where to place that marble. This right here is just like that promoter. Starting place, this is where you start, this is where you put it, right? And when, like in the example of the lac operon, when lactose is present, I'm going to try to pick this up. Then that repressor is lifted and we're able to make a copy, right? It opens up the DNA to make a copy, an mRNA copy that it can take to the ribosome and then go make whatever it needs to make. Does that help? Does that make sense? So today we spent a lot of time looking at pictures, talking about real life examples that are very similar to what we're doing today. That's because most of you probably never knew what a lac operon was. You probably didn't know what a promoter was, a repressor was. And one last example before you leave is kind of like glasses. Do any of you wear glasses? Yes. Some of you might need glasses to read. And that, like, if you don't have your glasses on, you can't focus on those letters or things are blurry. Yeah, you need them to see. So those are kind of like that transcription factor, kind of like that opener, right? It's like without those glasses, the repressor's on. You're like, I can't read that. But you put that on, and then all of a sudden you're able to read the DNA, right? It turns it on. It opens the door, and you're able to get a copy of that. All right. We have a quick check we did not get to go over today. So this is what we're going to do. We're going to go over this first thing tomorrow when you come in. We're going to start class. And then what that's going to do for us is that's going to help us rewind from today and reprocess it tomorrow, make it fresh. Okay. So how do you feel about what we accomplished today? Use your pointers. Do you now know what a lac operon is? An operon. Do you know what a promoter is? An operator is? Could you explain to me why Lizzie's lactose intolerant? Yes, look at all those yeses. Yes, yes, yes. Good. I don't know which one of those examples today stuck with you or made the most sense. That's why I gave you a variety. But what I want you to do is I want you to level this up. What does that mean? There is a quiz. I will change the due date till tomorrow at 9 o'clock. All right. So that we can go over that quick check together. You do get multiple attempts. So maybe you want to go take it right now and see how well you can do. And then if you didn't, wait till tomorrow when we review that, and then I'll allow maybe 5 minutes for you to pop in and check those. Okay. Yes, Lizzie, she can explain it. I love that. One of those examples might have stuck with you today. And this is what I want you to do before your next class, if you can, or in transition. I want you to get a piece of paper. I want you to get crayons, markers, a pencil. I don't care what it is, a pen that barely works. I want you to get something and write that down. I want you to make a flow chart or a diagram that explains this to you while it's fresh. Maybe it's the marble run. Okay, maybe it's the glasses. I don't know. Maybe it's the car wash. Whatever that is, write it down. If you just come to class, listen to me, and you're like, oh, that makes so much sense. And then you turn that off, right? You turn that off until tomorrow when you come back in. It's going to get foggy. It's not going to go from here on the triangle to here on the triangle, okay? So level up today. Do something to make this stick in your brain. Practice it. Make an example of it. Take that quiz. You got to go. I'll see you tomorrow morning, 8:30. We have help sessions. So if you want to go over this with me, pop into a help session with me, right? 8 o'clock before class, and we can get through this stuff. See, Lola, and that's might be why. That's probably why she gets sick. She's probably lactose intolerant, right? She's not producing those things that turn it on, fix the repressor and produces what she needs to break it down. Awesome. Have a great day, guys. I'll see you soon. If you have any questions, stick them in chat. Pop a hand up. Absolutely, Draven. Sadie, do you have a question?